My project is focused on an ultra-rapid molecular assay for the detection of pathogenic bacteria. And specifically, I'm focusing on E. coli and Pseudomonas aeruginosa. So just to give you a little bit of a background about the problem, infectious diseases driven by bacteria and viruses are the leading cause of disease and mortality in remote, isolated, and low-resource settings, and they have a significant burden on healthcare systems in those communities. And a prime example of this was during the COVID-19 pandemic in Canada. Northern communities with larger populations of Indigenous groups experienced significant delays in testing and tracing of the virus. And there are two primary reasons for this. One is that these communities are distant from centralized laboratories that have access to the standardized tests. And secondly, the standardized tests, the gold standard tests, which include PCR and culturing methods, take a long time for results. PCR is typically 3 to 4 hours, and overnight cultures tend to be 24 hours. So by the time you actually get your results, many days have passed. So the goal is to reduce the cost and time of the gold standard techniques while maintaining their sensitivity and accuracy in bacteria diagnosis. So for our proposed approach, we have developed an ultra-rapid molecular assay, which employs the loop-mediated isothermal amplification method, also known as LAMP, to detect E. coli and Pseudomonas within 30 minutes. And the biology of this is that as the DNA of these bacteria amplifies, as nucleotides get added, H+ ions are released. And as a result, the reaction changes from a more basic pH to an acidic pH. And we're able to detect this using a pH indicator, which in our case is phenol red. And the reaction changes color from a fuchsia to an orange or yellow in a LAMP reaction. And we also noticed color changes when this assay was integrated into a microfluidic device with color changes within 10 minutes. So for the methods, the first aim was to design the primers that we would use. And this was based on studies that have been already published with high-performing primers that can detect color changes in 60 or 70 minutes off chip. So for E. coli, I've displayed the set of primers I use, and same for Pseudomonas. And for the LAMP reaction itself, there are 4 main components. The first is the colorimetric master mix, which contains the DNA polymerase that's responsible for amplifying the DNA of interest. We have the primer mix, DNase-free H2O, and of course our DNA sample. And to validate our primers in a simple LAMP reaction outside of the microfluidic device, I ran a simple water bath with a temperature range of 60 to 65 degrees Celsius, which is the optimal range for a LAMP reaction to occur. And for E. coli DNA, I noticed color changes within 20 minutes using our reaction for a concentration of 70 nanograms per microliter. And for Pseudomonas, I noticed color changes after 30 minutes. And then we confirmed our results in our microfluidic device. So if you look at the results section, for E. coli, we tested various concentrations across 60 minutes, and we noticed color changes for every single concentration that we measured within 60 minutes. And we also did a quantitative analysis of our microfluidic device using a microscope. So once we took our microscope images, we analyzed the RGB values of the images that we obtained. And specifically, we compared the blue and green values of those images from the microscope. And what we observed was that within 10 minutes, we noticed a decrease in the ratio of blue to green from a 10-to-1 ratio to a 2-to-1 ratio. So within 10 minutes, we were able to quantitatively observe that the ratio of the colors was decreasing and therefore changing. And we observed the same trend for our Pseudomonas samples. So within 10 minutes, we noticed the blue to green ratio significantly decrease in the microfluidic device from a 10-to-1 to a 2-to-1 ratio. So overall, this method is significantly more rapid compared to the 3 to 4 hour PCR or the overnight cell culturing methods that are typically used for bacteria diagnosis. And combined with microfluidics, we were able to receive results within 10 minutes. And I think that with this approach, healthcare systems in low resource settings will be better prepared to combat future infectious disease outbreaks. So the next steps are to test this assay with a gram-positive bacteria like Staphylococcus, and hopefully this approach can show a proof of concept for an optimized molecular assay for bacteria diagnosis in healthcare settings. So I'm open to questions if anyone has any.
Dr. Bai, do you have any questions?
Yeah, so I have two questions. First is like you mentioned about this is a very good way to as a point of care diagnostics, right? But perhaps I missed it. Did you show like you didn't show like a diagram of the microfluidic device? So
Right.
my question is like, are you able to incorporate, a temperature control on your chip, and also incorporate like a ways to lyse the cells, to get the DNA in order to do the lamp amplification? So is that something that you can do on the chip?
Yes, so we actually have a heater element that we connect to the microfluidic device. And the reason I didn't show that was actually because I wanted to focus more on the assay. But we actually are able to obtain temperature ranges in 60 to 65 degrees Celsius for the lamp reaction. And for the lysis, we were actually able to have the heater element, the chip, up to 95 degrees Celsius. So we first lysed the samples on chip and we extracted the DNA, and then we ran the lamp experiment in the device. Yes.
Cool. And then another question is, you mentioned about your, the choice of those primers based on like a previous literature, right, as a benchmark.
Mm-hmm.
Have you thought about, better optimize the primer design so that you can possibly detect multiple serotypes or multiple strains?
That's a really good question. So actually, this was actually my second set of primers that I tested. These were optimized based on a failed experiment in the past. And basically, the reason we're choosing these primers is because they have high specificity for the specific bacteria we're interested in. So if we were to use primers that were general to more than one bacteria strain, it would be a trade-off with the specificity for the bacteria you're interested in testing for our case.
Cool, thank you.
Thank you for your questions.
I got a very quick question. I've been working with the detection of bacteria in the past. So my question is that, can you, with this technique, can you do a multiplex in terms of multiple bacteria at a given time, one sample?
Yes, exactly. So actually our device itself, which again, I didn't show, but it has multiple chambers. So we're actually able to run 3 experiments at a time. And we can do those simultaneously because this assay essentially requires the same temperature range for the lamp reaction across multiple bacteria. So yes, we can run more than one experiment at a time.
Dr. Gorbet?
Yeah, thank you. Yeah, it would have been nice to see the device because I know I had trouble understanding the on-chip all the time where I didn't see the chip.
Yeah, exactly.
Yeah, so it makes it a little bit difficult, just so that you know. So looking a little bit ahead, and it wasn't clear to me also. So thinking about a point of care device, currently, your samples are likely clean samples in a way. So how do you expect that might actually, what do you anticipate some of the challenge you may have in, getting that to be a point of care or a diagnostic tool?
Absolutely, that's a really good question. So with point of care, I think the trouble can be the sample you use, because one concern that I have right now is if we're testing blood samples, the blood color might actually interfere with the phenol red change. So I think in a point of care setting, the goal is to use saliva or urine samples that are a little bit more transparent so you can observe the color change. So I think in the point of care application, the difficulty would be to specify which sample is best for a given bacteria. However, not all bacteria can be detected in a urine or saliva sample. So that's kind of the difficulty and challenge I'm actually encountering now, and hopefully troubleshooting that for the next steps of my research.
Great, thank you. That's really neat work. Thanks.
Thank you.